Okay, so next up, we have Dr. Ethan Ahler, who recently completed his graduate work in the Fowler Lab and is currently a scientist at Revolution Medicines, which is an oncology drug discovery company in the San Francisco Bay Area. And Ethan will talk us through picking a model system today.
Great. Thanks, Sean, for that introduction. So today I'm going to talk about some of the broad technical and biological considerations you should have when trying to make a choice for a model system for a deep mutational scan. And then I'm also going to go a little bit into some anecdotes about DMSs that I've performed in both yeast and mammalian cells. So really the fundamental purpose of a model system in a DMS is that it serves as that physical linkage between the variant protein that's being expressed and its underlying genetic sequence. And that's the case whether you're working in mammalian, virus, or yeast. But I think the model system choice has a lot of implications far beyond that sort of fundamental role. And so not only is it one of the very first choices you have to make before doing the DMS, I think it really impacts every layer of the traditional deep mutational scanning process, all the way from determining what types of functional assays you have available, including which ones are even possible. When you actually do your scan, it can inform and constrain really how you get your variant library into the cells, how you express that variant library, and then how you get the DNA to sequence out of them. And then finally, I really think it's it serves as a lens through which the results of your DMS must be interpreted. So basically, any biological or technical caveats that are true for your model will sort of be carried through towards all of your DMS results. So right now, or here in this figure, I'm just plotting the number of DMSs by year that have been completed that I pulled from PubMed, and this is definitely not a complete list. But I've colored them according to the model organism that they were performed in. And kind of as you can see here, the diversity of DMSs that have been done have also been performed in a wide variety of model systems. And there really doesn't seem to be any hard and fast rules about determining which proteins are suitable for which model systems. So I think there's a lot of flexibility inherent in this, which leads us to the question, okay, how do we go about picking the right model system for our particular deep mutational scan? And for me, really the fundamental thing I think about is you're in this balance between the biological rationale for sort of, is your gene going to function the way you think it is in your model system? And that almost always comes with the trade-off of technical tractability. Like how easy is it going to be to perform the scan, which, as we know, when we scale up to these massively parallel approaches, normally easy things for single variants can quickly become very challenging and serve as a large barrier in some cases. So I'll start with some of the questions I think about on the biological rationale side of the model selection. So one really easy question to first think about is, as you're deciding between model systems, is the species of the gene that you're studying, is it the same as the model system that you'll be expressing the gene in? And if the answer is yes, like on the case on the left, then I think it's pretty straightforward. The biological rationale is pretty straightforward, right? But oftentimes, for technical reasons, we have a case on the right where we have to express our protein of interest in a system that it did not originate from, aka heterologous expression. And I actually don't think, and this is a huge issue in many cases, and I can actually think of some instances where this type is an advantage. But I do think when you are doing a heterologous expression model, you kind of have to think more carefully about the biological implications. So for example, some questions I've thought of, if you do take this type of approach, is your protein going to have the correct subcellular localization for its function? If your protein is dependent on interacting with one or more other partners, are they present in that model at appropriate levels? Many proteins require post-translational modifications for their correct function. And you need to make sure that the machinery to do that is present in your model. And this is a personal example for me. So early on in one of my scans, we were deciding between E. coli or yeast. And I didn't realize at the time, but the protein I was studying really required a post-translational modification, myrcillation, which is only present in eukaryotic systems. So I was kind of barking down the wrong tree, and I was the first year, so I had no idea that that was even a problem. And then just more broadly, are the cellular systems in place in your model that your functional assay depend on? So another easy example is if you're doing a splicing assay, then you probably don't want to do it in a prokaryotic system. And you can think of many of these other case examples as you go. Kind of related to this question is, okay, once you finally decide on the species of your model system, you're often faced with the task of, okay, which specific cell type or which specific strain do we actually want to go for? And this is very apparent when you're looking at all the hundreds of different human cancer cell lines that you could choose to do your DMS in. And again, oftentimes when you stray away from the more common workhorse cell lines, technical issues that you've optimized in those become much, much more trickier. And so it's this balance between biological, the appropriateness of the model, and then how easy it is to actually use it. And my personal view of this is that if your functional assay is kind of more protein intrinsic, for example, you're measuring abundance of a variant, then maybe a more general cellular model is appropriate. But if the function is something that's going to be pretty specific to your cell type, like if you're looking at cis regulatory elements, then maybe the technical challenges that come with using a model that's more disease or cell line specific are probably going to be worth it. And related to this, I just want to bring up this quote from a recent publication by some former colleagues of mine, where they point out that many of the DMSs that have been done are either non-human cell lines, and this is with a clinical focus, or either non-human systems, or are in these non-necessarily most relevant for disease models. And despite that fact, if you correlate the effects from these deep mutational scans with known pathogenic variants that have been determined through other measurements, there's actually a pretty good correlation. And so I think that's, you know, for your specific example, you're obviously going to have to take it case by case. But I think in general, at least, there's evidence for that you can get really clinically applicable results using non-disease relevant models necessarily. So beyond that, I think it's actually pretty rare once you finally choose your model system that it's ready to go right out of the box. You're going to have to, in many cases, make additional modifications in order for either the biology to be more relevant for your DMS or for technical reasons. So very straightforwardly, you can do genetic manipulations to your model. One thing I think is fairly common to do is if you're expressing a gene, you might want to CRISPR out the endogenous copy of that gene, just in case that the wild type is masking any effects you might see from your variant that you're expressing. You can also co-express additional partner proteins. Again, if your gene requires functional interactions with other proteins. And then there are also some more environmental modifications you can do to your system, like adding drugs or changing the culture conditions to either improve the technical or biological readout. And this is, as I've learned from personal experience, this can be a very time-consuming thing. So I think it really behooves you to think about what modifications are really necessary and the validations that go into it. And so just to give some real world examples of modifications that people have done in order to enable a deep mutational scan, I think there's a really nice example from Clara in the Dunham lab, who spoke yesterday. So she did the DMS on a human protein, CYP2C9, and she performed it in yeast. And what she noticed is that if she just expressed CYP2C9 by itself, she didn't get a good readout in her functional assay. And it kind of makes sense because in human cells, CYP2C9 activity requires the function of two other human proteins, B5 and CPR, for full activity. And so when she humanized her yeast system by expressing these other human protein factors, as you can see on the graph to the left, she was able to recapitulate a really nice activity of the CYP2C9. As another example, other groups have taken protein phenotypes that might not necessarily be so easy to read out in the cellular system, like protein aggregation or lipid phosphatase activity, and have, through pretty clever engineering strategies, I think, converted these functional readouts into a yeast growth assay, which is a very straightforward functional selection to work with. So I suggest you take a look at these publications for the details and how they did this. But they involved either adding drugs or co-expressing with different human proteins. Okay, so going back to more considerations to think about when making your model selection, and this is more on the technical side of things. One really big thing that stands out when you're comparing different models is trying to get your library of variants into a model. So generally, mammalian cells are sort of capped on the amount of variants you can easily introduce into them at around the 10 to the 3, 10 to the 4 range. Whereas for models like yeast or E. coli, you can get quite a few more variants in much more straightforwardly. And so when you're thinking about, okay, I want to do a DMS of this gene, it's going to be this long or this many amino acids. And so you can calculate the theoretical size of your protein. And based on that, it might help inform which model you end up going just for technical reasons. But as I said, these are pretty rough numbers because as technology improves and depending on your specific model, you can see different values for these. Kind of related is this sort of fundamental rule in demutational scans where you really need to maintain, in order to get that genotype-phenotype linkage, you need to maintain this 1 variant per cell rule. And this can range from being pretty straightforward to being pretty tricky. So for example, in yeast and bacteria, a common way to get your library into cells is through plasmid transformation. Now, E. coli, it's, from what I've read, multiple plasmid transformation is a pretty rare event. But in yeast, depending on the method you use, it can be an extremely high percentage of cells will take up multiple plasmids, i.e. multiple variants. And that will obviously not be good for your readout. So I strongly recommend including controls in your transformation protocols to assess the rate of multiple transformants. But then also, at least with certain protocols, one paper noted that you can ameliorate the multiple plasmid transformation problem by just doing a really extended outgrowth, and the plasmids will segregate out during the growth. For mammalian cells, using the most common form of library introduction, which is lentiviral transduction, you really need to be using a low MOI in order to ensure or bias that a cell will only pick up a single viral particle. And as just an example for some real numbers, at an MOI of 1, so 1 virus per cell in your infection, about 26% or a quarter of cells are expected to receive more than 1 variant. Whereas if you do a much lower MOI of 0.1, that number of cells quickly becomes quite negligible. So again, these are parameters you have to optimize in your own. And so to wrap up, I'm just going to go through some real case examples of 2 DMSs I've worked on and explain sort of the rationale why I chose the models that I did. So for my first one, I wanted to do a deep mutational scan to understand the sequence determinants that regulates catalytic activity of a human kinase, which is called SARC. And I ended up picking yeast as my model. And the reason I did yeast was several reasons. First of all, there was this extremely well-established functional assay for SARC activity in yeast, and it was based on growth. And literally, it was first described decades ago, and many groups have used the same assay specifically for SARC. And this was extremely valuable. A, it gave us a lot of confidence that I could get this working because many other groups have gotten this working. But B, really, each publication that used this assay kind of provided more evidence that doing a human protein in a yeast model accurately reflected the human biology. So that really saved us a lot of time in trying to justify the case for studying a human protein in yeast. The second thing is that given our functional selection was growth-based, it's really nice to for yeast because they have such a quick generation time or low doubling time. And really this allowed us to do both pilot experiments and the actual selection experiments in a matter of days rather than weeks like you would see in mammalian cells. And then finally, I think initially I thought this might have been a downside to the system, but now the more I thought about it, I think it's actually a benefit, is that yeast lack the normal complement of regulatory proteins that regulate SARC activity that you see in mammalian cells. And so what this means is by stripping away all of the systems of regulation that you would find in mammalian cells, all of the effects you're seeing, we think, are coming sort of solely from the protein intrinsic effects of the mutation. And that can really sort of clean up the data that you end up generating. But finally, but even with this really well-established model, I did have to make several optimizations in order to get this working for my specific deep mutational scan. So in addition to being interested in understanding how mutations affect catalytic activity of the kinase, I wanted to understand if they affected inhibitor resistance when I treated it with a kinase inhibitor. But one downside to yeast is that there's this really well-known phenomenon where they're generally much more resistant to drugs than mammalian cells are. And so to get around this, and this is due to both, they have quite a few drug efflux proteins, as well as having a less permeable cell membrane. And we actually used, Fritz Roth in his group actually generated a strain of yeast where they knocked out every single drug efflux protein in that yeast strain and noticed that it caused, it sensitized yeast to quite a few compounds. And indeed, when we tried it in our hands, we were finally able to get some of our kinase inhibitors working. But that came with a cost because this strain was much more slow growing and sickly than the parental strain. And it actually resulted in about 10-fold less transformation efficiency. So it took optimizations in order to get that transformation efficiency back to a level that I could use for my library. And I think in the interest of time, I'm not going to go over the mammalian cell DMS experiment that I've done, but I'd be happy to talk about this at lunch if anyone's interested. And so I will wrap up. Just say that I think some of the really major points you should do when thinking about which model system to choose is to read both really deeply and broadly in the literature. I would say a lot of the key papers I found that made me eventually decide on the model system I chose came from way back ago, where they were able to prove that the model system it's using would be valid for the protein as it should be. Once you figure out that your model is biologically suited to your protein, then I think you should really do as much as you can to make this technically feasible on the onset, just because, as we all know, doing deep mutational scans can be very challenging. And so you want to set yourself up for success. I think it's okay. I think you can rationalize all these caveats. I think that's sort of the trade-off you get when you're doing these massively parallel assays. But I think it's good just to keep track of all these caveats and then perhaps design validation experiments later down the line that can address the caveats that you have in mind. And then finally, I think it's quite clear that in many cases there is no one best model, that model selection can be very subjective. So a lab might pick a model because they have prior expertise in it or they have access to reagents. So I think as long as you do your due diligence for your given model, in a lot of cases, the model can be a perfect answer. And with that, I'd be happy to take any questions.